tumor cell lines, we have established more than 20 cancer disease models, which can be categorized into 6 major types, including syngeneic models, CDX models, xenograft models. Please note, PDX model is not available at ProBio due to the biohazard safety regulations. These models are widely used in drug candidates screenings and validation. To facilitate the analysis of biological samples and comprehensive evaluation of developabilities, a variety of platforms available at ProBio, including ELISA-based PK, cytokine, and biomarker profiling services, fax-based oral and typing, including CAR T cell detection services, and also pathology and RH-based analysis. To help everyone get a better understanding of the overall process, here is a high-level flowchart demonstrating the scope of analytical work. The animal tissue samples is separated into serum and the tumor tissues. The serum and the plasma will be directly processed for in vivo PK and cytokine analysis. Alternatively, the plasma from unchallenged animals can be used for ex vivo stability tests. On the other hand, the tumor tissue will be sent to cell profiling or preparation of FFPE for pathology studies. Given the complexity of a preclinical animal-based studies, in this talk, we will only focus on the PK part, the rest of the presentation. Prior to the animal dosing and sampling process, generation of critical reagents such as anti-idiotype type antibodies and the PK assay development needs to be conducted and well validated. We will cover this part in the second and the third section. In rodent-based PK studies, one of the main concerns is that the human IgG Fc from the drug does not associate with mouse FcRn, the Fc receptor, as strong as human FcRn. Since the interaction between Fc and FcRn leads to the recycling of the drug, resulting in extended half-life. This weaker interaction between human Fc and mouse FcRn brings a pre-existing bias in PK profilings. So to solve this issue, human FcRn transgenic rodents were generated to better mimic the human immune systems. In this human FcRn transgenic mouse model, expression of a human FcRn is first confirmed at mRNA protein levels. This model is particularly useful when it comes to PK profiling with Fc mutations that are supposed to extend half-life, such as the YTE triple mutations. So the YTE triple mutation in human IgG Fc is known to enhance the binding affinity towards the FcRn, the Fc receptor, leading to better recycle of the drug and improve the PK profile. So in this study on the right panel, the antibody candidates with YT triple mutations demonstrated the red curve demonstrated lower drug degradation and longer half-life. If the same experiment is done on Y-type mice, you might not be able to notice this kind of difference. Transgenic rat with the same knocking of a human FcRn is also available in the market, and similar results is observed on the right panels, in which the human IgG-YT mutation and the N434A mutation demonstrates better PK profile comparing with the wild type. The knocking of human FcRn resolves one issue, the one I mentioned previously, but it also brings another problem onto the table. HSA is the natural ligand of FcRns, and it also showed FcRn affinity dependent PK profile, which means the HSA variant with higher binding affinity towards FcRn tended to have a longer half-life, while low binders comes with shorter half-life. In the FcRn transgenic mice, the mouse HSA has a higher affinity towards human FcRn transgene comparing to human HSA. As a result, the mouse HSA will occupy human FcRn to a higher percentage and reduce the drug IgG binding to FcRn, leading to another bias in the PK profile. To resolve this new problem, HSA and FcRn double knocking mouse is generated. So in addition to the in vivo animal services, ProBio also offers comprehensive one-stop solution from lead discovery and engineering to pharmacology characterizations, including in vivo and in vivo PK toxic studies. Such integrated platforms will greatly reduce the overall timelines by conducting multiple milestones in parallel. The typical timeline from target evaluation to clinical candidates is about 12 months at ProBio, as compared to almost 2 years if you choose to do this work step by step. So we're hoping that our expertise can help accelerate your preclinical development. Next part, we will moving on to the second session, the application of anti-idiotype antibody for PK studies. As I mentioned previously, anti-idiotype antibody serves as a critical reagent in PK and toxicity studies. It is a type of anti-drug antibodies that can specifically recognize the variable region of each drug. So this kind of specificities enables the anti-idiotype antibody to become a useful tools to measure the drug concentration in serum samples, which is the PK studies. Alternatively, it can serve as palliative control in the ADA studies to measure the total level of ADA, the anti-drug antibody, or the level of a neutralizing antibody, neutralizing ADA. Typically, there are several different assay formats available for PK assay development. Antigen capture is commonly used in discovery stage since it is relatively cheap and easy to set up without the requirement of anti-idiotype antibody generations. The recombinant protein of the drug target is directly coded on the plate to capture the drug in the serum samples, followed by the detection with the commercially available common anti-Fc secondary antibody. Universal PK2 antibody is exclusively developed by ProBio's team and demonstrated decent detection matrix as the other options. We will have detailed technical discussion in the last part regarding the universal PK2 antibody. Anti-idiotype capture and anti-idiotype sandwich ELISA format both requires anti-ID, anti-idiotype antibody coded on the plate to capture the drugs in the supernatant, followed by detection with either commercially available anti-Fc secondary antibodies or another anti-idiotype antibody with different epitope. Generally speaking, anti-ID sandwich ELISA format comes with the best detection metrics, while universal PK2 antibodies comes with similar performance, but it can only be used in animal-based study, but not human samples in clinical trial. Antigen capture approach is the most cost-effective approach with the sacrifice of detection sensitivity and accuracy. The sandwich ELISA formats can be adjusted or manipulated based on different drug modalities. For instance, to detect intact ADC drugs, one anti-idiotype antibody is coded on the plate as a capture reagent, while another antibody specifically recognize the small molecule toxins comes in as the detection reagent. Any half-degraded ADC will not be detected or included in the PK profile. Likewise, to detect the intact bispecific antibodies, a pair of anti-idiotype antibodies specifically recognize each end of the bispecific will be used to measure the intact drug and exclude any degraded parts. To establish sandwich ELISA, the first step is to identify antibodies, anti-idiotype antibodies that can potentially form a pair, which means these two antibodies can associate with the drug simultaneously without any competition in between. So in this checkboard ELISA, 10 anti-idiotype antibody candidates are mapped with each other to form a 10 by 10 array. And the drug in between is set up as a 4 serial dilution 0, 1, 10, and 100 nanograms per milliliter for each pair. Based on this ELISA profile, the pairs with the highest signal at the lowest concentration is considered as top pick, as the one showing here in highlight as green. After the selection of the top pick, the top pair, a further sandwich ELISA with more refined drug serial dilution is conducted to establish the standard curves and the detection range. The dilution range is from 0 to 20 nanograms per milliliter. So here is a list of typical metrics for PKS development. Besides the standard curves and the detection sensitivities mentioned in previous pages, the assay precisions, the spike recoveries, dilution linearities, and stabilities are also crucial parameters. Next, let's move on to the last part, the introduction of the universal PK2 antibody. So, although, as I introduced in the second part, although the anti-idiotype antibody is the golden standard for PK studies, it takes quite some time to generate these reagents, and sometimes you may not succeed in finding such a good pair of anti-idiotype antibody. Moreover, if you want to run preliminary PK study at the discovery stage, it is impossible to generate anti-ID antibodies since you need the tox batch from the CMC stage as the immunogen to generate the anti-ID antibody. So under these circumstances, ProBio developed a panel of a universal PK2 antibodies. These antibodies specifically recognize the constant region of a human IgG with different isotypes, and it has minimal cross-reactivity towards the IgG from regular lab animals such as mice, rats, rabbits, cynomonkey, dogs, etc. So this ready-to-use toolbox enables a quick and easy PK profiling during the late discovery stage and helps you making the decision about the selection of candidates for the downstream cell line generation and CMC process development. Comparing to anti-idiotype antibodies, the universal PK2 antibody is already validated for pairing and ready to use for PK assay development. However, given the fact that it recognizes all human IgG, it can only be used in animal-based PK study, but not in clinical trial stage with human patient samples because it cannot differentiate between the background human IgG in human series versus the drug IgG in the serum samples. Comparing to the commercially available catalog secondary antibodies, universal PK2 antibody greatly improves the specificity and reduces the matrix effect. Comparing to antigen capture PK ELISA, which is currently the most well accepted, the most popular approach for PK study in discovery stage, universal PK2 antibody not only outperforms in terms of the assay metrics comparing to the antigen capture approach, but also eliminates the restriction on the availability of recombinant antigens. For instance, if you have a GPCR antibody drug candidate, it is almost impossible to run antigen capture PK ELISA because there's just no recombinant GPCR protein available with decent qualities. So this problem will no longer exist with the universal PK2 antibody. So next, I will share some validation data with this universal 2 antibodies. First, it is the ELISA titration experiment against various kind of IgGs. So this data demonstrate that these 2 antibodies associate with human IgG, which comes with different isotype, IgG1, IgG2, IgG4, and different modalities, the monoclonal antibodies and the ADC antibodies uh, at the nanomolar affinity range. In the meantime, these 2 antibodies does not cross-react towards any other animal IgGs, including rats, dogs, cynomonkeys, and rabbits. So among these 2 antibody panels, we have already identified several pairs uh, that shows decent detection sensitivities and can be used directly for scavenger ELISA assay development. In this checkboard ELISA, clone 17 and 18 acts as capture antibodies, while clone 4 and clone 5 with biotinylations serves as detection antibodies. By doing the spike recovery test, this sandwich ELISA PKSC format demonstrated excellent standard curve consistency between 3 independent experiments. In this test, we are comparing the nominal concentration, which is the actual drug concentration added in the samples versus the, the measured concentration, which is calculated with the standard curves. The differences in between the nominal concentration versus the measured or the calculated concentration is well controlled under 10%. And the majority of this sampling data is as low as roughly around 1 to 2% of variations. Based on the standard curve, the detection limit can be calculated by average of 24 samples and 4 sets of data. For this particular antibody pair, the detection limits can reach as low as 10 picogram per milliliter. In addition to accuracy and sensitivities, these universal 2 antibodies also show great tolerance under the influence of the matrix effect from different animal series, such as cynomonkeys, and dogs, and rats, and the mouse. The standard curves remains acceptable range of variations when exposed to these animal series with 1 to 10 to 1 to 50 dilution ratios. The typical acceptable dilution range, MTD, for animal serums is from 1 to 10 to 1 to 100. So this dilution ratio is well falls into the acceptable range. Okay, so that's pretty much everything I have for this presentations. Let's move on to the Q&A session.
Thanks for Dr. Chen's excellent presentation. If you have any questions, please type it on the chat or Q&A panel. As you enter the questions, please also follow our LinkedIn account, ProBio, and the Korean audience can also scan this QR code and follow our Kakao channel. There will be more online sharing resources, and we will share them on our LinkedIn and the Kakao channel at the first time. Okay, Dr. Chen, I received one question. The question is, how about the LLOQ? Could you please answer it? Yeah.
Yeah, I think we can address this question offline to emails regarding the question about the LLOQ.
Okay, sure, no problem. Okay. Okay, and here the second question is, do you recommend mouse or rabbit immunization for the generation of anti-ID app?
Typically depends on what you're doing for the PK or for the ADA. Usually I would recommend to the rapid immunization and collect the polyclonal antibodies for the ADA assays as the part of control. While for PK assays, people usually use the mouse immunization plus either hybridoma or phage to screen for monoclonal anti-EDO antibodies. I mean, you can also use the rapid versions, but usually the mouse version is more than enough.
Okay. Okay, thank you for your answer. Here is another question coming. Is it necessary to run preliminary PK in rodent?
In recent years, more and more people start considering preliminary or pilot PK studies in late discovery stage, primarily because the cost for the animals is much cheaper than previously. So the answer is yes. I would suggest to run a preliminary or pilot PK studies in mouse or even rats, just to give a to share some insights into the metabolism of your candidates. It will help you to make the decision about which candidates is giving a better PK profiling, and which candidates is more suitable to move forward with CMC. But one thing to note that the rodent-based PK profiling is definitely not equal to the monkey-based or the human-based study. It's even with the FCRN transgenic animal, the FCRN transgenic rodent, it's not necessarily showing the same profiling. It's only a way to help you narrow down candidates.
Okay, got it. Very clear. Yeah. And the following question is, which assay format do you recommend for the PK assay?
So, as I mentioned in presentations, anti-EDO type antibody with the 7G ELISA is definitely the top choice. But some of the time, you may not be able to find a pair of anti-AD that can form a complex with the drugs with each other. So sometimes people will take the next best options to go either with the anti-etotype capture, in which you only need one good anti-etotype, but not a pair. If you are considering doing the preliminary PK studies in discovery stage, it used to be the antigen capture approach used to be the only option because you cannot generate anti-etotype at this stage. But nowadays we have the universal PK2 antibodies. It can definitely help you to run a better PK profiling work at discovery stage. Definitely much better than antigen capture approach.
Thank you. Thank you for your sharing. That's all the questions I got from my side. If there is no more questions, we'll close this webinar by now. And here, I also want to introduce you or another webinar we are going to have on the 4th March. The topic is about the multiple solutions of functional leads discovery and engineering. If you are interested in it, please kindly find more details through our link in the chat box. If you have any more questions, please feel free to contact us through our official email. Let me join others in thanking Dr. Duet Chen for his time today. And thank you all for attending today's webinar. Look forward to meeting you soon again. Thank you. Bye.
Thank you, everyone. Have a good time.
You too. Bye.